Hey everyone, this is Vaibhav Shandelia from ESIC Medical College, Hyderabad, and today we'll be talking about a very easy organism which is a fungus, and it's called Histoplasma Capsulatum. Let's start with Histoplasma Capsulatum, and coming to the introduction, this is a systemic granulomatous disease. This is what you'll write in the definition: a systemic granulomatous disease. And contrary to what it sounds like, this organism is not capsulated. So it's a misnomer. It is not capsulated. You need to remember that. And it's also known as Darling's disease after the person who first reported it, Samuel Darling. This is a dimorphic fungus, and we'll learn about this further in the lab diagnosis, what this means actually. And also, something important for the MCQs is that this fungus inhabits in the humid and acidic soil containing bird and bat droppings. This is very important. The moment you see the word bats in a clinical vignette, you need to first think about histoplasma. Okay, histoplasma and bats go hand in hand. Okay, after the introduction, let's start with the pathogenesis. So this organism, it spreads through the inhalation of spores, and the spores are basically the microconidia. So there is inhalation of spores and they go into the lungs. In the lungs, they are ingested by the alveolar macrophages, and inside the macrophages, these microconidia, they transform into yeast form. Okay, the microconidia or the spores transform into the yeast form inside the alveolar macrophages in the lungs. There, they survive within this now called phagolysosome. They survive within these macrophages using secreting bicarbonate and ammonia. Secrete these two things and survive. The yeast is surviving. After this, they multiply inside the phagolysosome, and then they spread to the regional lymph nodes and bloodstreams. Okay, they get out of the phagolysosome, they burst out and spread through the regional lymph nodes and the bloodstream and spread to other parts, and as they are spreading throughout the body, they trigger the cell mediated immunity. Now the cell mediated immunity is triggered, and what this leads to is the formation of a granuloma, and this is the most characteristic feature because we saw in the definition this is a systemic granulomatous disease. Systemic because it spreads to many parts of the body, right? And granulomatous because we saw cell mediated immunity triggering granuloma formation. So there is granuloma formation, and as this granuloma heals, this leads to calcification and fibrosis. Okay, calcification and fibrosis. One thing to remember is this is the end stage of the disease. There will be no reinfection, that does not occur here. As soon as it heals, granuloma forms, there is healing, calcification, fibrosis, end of disease. After pathogenesis, let's go to the clinical manifestations. So, if a person has a strong cell-mediated immunity, right? If they have a strong immunity, they'll mostly be asymptomatic, right? They'll mostly be asymptomatic. Otherwise, we can see three manifestations. Firstly, pulmonary histoplasmosis. Okay, pulmonary histoplasmosis has two acute and chronic forms. The acute forms shows us a flu-like illness. There is infiltration seen in the chest X-ray, hilar mediastinal lymphadenopathy. Okay, this is what is seen with the acute form. Basically a very general flu-like illness. In the flu, we can also see infiltration in the chest X-ray and lymphadenopathy. We'll see all that here in the acute form. In the chronic form, however, on the X-ray we can see cavitary lesions. Okay, a little bit more severe, we can see cavitary lesions. Coming to the mucocutaneous histoplasmosis, the second manifestation, this occurs almost always secondary to a pulmonary histoplasmosis. That means first you'll get the pulmonary disease followed by a mucocutaneous always. It never occurs that there's a mucocutaneous manifestation. It's always a pulmonary followed by a mucocutaneous. And here we can see skin and oral mucosal lesions. Alright. Finally, we have a disseminated histoplasmosis, which occurs when the cell-mediated immunity is very low. And the disease will now spread to the bone marrow, spleen, liver, eyes and adrenal glands, causing more severe manifestations. Okay, so these are the three: pulmonary, mucocutaneous and disseminated. However, if you have a strong immunity, you'll mostly go asymptomatic. Coming to the lab diagnosis, you take a specimen for analysis from the sputum. You take an aspirate, bone marrow, lymph node, blood, or biopsy of the skin and mucosa. Remember the lesions, mucocutaneous lesions, you can biopsy those. Coming to direct microscopy, we have PAS, GMSA, Gomori methenamine stain, and all these will characteristically detect the yeast form, the oval yeast cells, right? And what we can see is these yeast cells budding within the macrophages. Remember, they were surviving using the bicarbonate and ammonia. So you can see that within the macrophages, you can see them surviving, the yeast surviving and budding within the macrophages, and you can also see a granulomatous response under microscopy. Cool. Next, coming to the culture, this is a gold standard. Culture is the gold standard, and we culture under Savarod's dextrose agar, blood agar, BHI broth. And this is where the dimorphic fungi comes in. This is why we call dimorphic, because this fungus can exist in two forms at two different temperatures. Very important, at 25 degrees Celsius and at 37 degrees Celsius. So at this colder temperature, the 25 degrees Celsius temperature, we see a mold form or a mycelial form. Okay, so you'll see this mold form, which basically forms white, brown, buff colonies, characteristic white, brown, buff colonies. And this is basically consisting of the tuberculate macroconidia or the microconidia, right? Microconidia. Remember the ones which were infecting the body first? This is the mold form. Secondary, we see at 37 degrees Celsius, more hotter form, we can see the yeast form. From the mold, we get to the yeast form, and this grows best in the Kelly's media. A very simple mnemonic to remember this would be yeast in the heat and mold in the cold. Okay, clearly yeast in the heat and mold in the cold. A very easy mnemonic to help you remember. Next, we can get antibody detection by CFT, immunodiffusion test, and skin test. We can do a hypersensitivity reaction to the histoplasmin antigen can be detected, and again molecular methods will be PCR. The treatment for this disease will be amphotericin B for the acute form and itraconazole for the chronic form. Alright, so a very easy organism, Histoplasma capsulatum, very short. Some highlights: it's also known as Darling's disease. It is not capsulated, it is a granulomatous disease, and it's dimorphic. You can link it with pat droppings. And coming to the pathogenesis, it goes inside the macrophages via the lungs. These spores go inside, form the yeast. There they survive, multiply, spread to the lymph nodes and bloodstream, spread to other parts, cell-mediated immunity triggered, granuloma forms. Clinical manifestations, we have three: pulmonary, mucocutaneous, disseminated. First is usually pulmonary, leads to mucocutaneous. If you're too weak, you'll get disseminated, and if you're strong, you'll probably stay asymptomatic. Lab diagnosis, the things you have to remember is, under microscopy, you'll definitely see yeast cells which are budding within the macrophages, and you'll see a granulomatous response. Under culture, you'll see two forms. The most important point you can take from this video: yeast in the heat, mold in the cold. 25 degrees Celsius mold form, 37 degrees Celsius yeast form. The mold has white, brown, buff colonies. Okay, and that's probably it. Nothing much to remember. And yeah, see you guys in the next video.